On today's episode of OMG OMX, you're going to hear from Mike Greig, who's the director of Bruker's Pharma Biopharma division. I hope that you enjoy his story. He's got some really interesting anecdotes that I think you'll find enjoyable. Hi, Mike. I'm excited to welcome you as a guest to Bruker's OMG OMX podcast series. As usual, I was hoping you'd start by sharing your personal story with us and taking us through your journey of how you got to Bruker.
Okay. So I started out at University of Delaware. I was a chemistry major there. I was born in Oregon, went to high school outside Chicago, so I thought I'd try the East Coast for college. So I went there. After Delaware, I decided to head back west. And I went to San Diego and I got a job with Revlon. Revlon had a very small lab in San Diego. And there I was, got a job as a polymer chemist doing nail polish and lipstick. So that was quite exciting. I did that for about a year and a half. And then I decided I would try something different. And so I went to work for a biotech company called Isis Pharmaceuticals. It's an antisense company. They're called Ionis now for obvious reasons. So I started there, and that's kind of where I was first exposed to mass spec. We got a mass spec at ISIS, and we had 3 of us volunteered to try to learn how to use it. Fortunately, I could figure it out pretty quickly. The very first things I ever did on a mass spec were oligonucleotides, doing negative mode, and that's the only world I know. I didn't know most of the world did positive mode, and thought negative mode was not so easy. So I got a good start there. The very first paper I ever wrote was a native mass spec paper, but we actually didn't even have native mass spec in the keywords because we didn't know that it was even a term at the moment. While I was there, I met a bunch of scientists from Alan Marshall's lab at the National High Magnetic Field Lab at Florida State. And I was fortunate to spend time there as a visiting scientist. So I did a lot of ICR work. And I think the experience in Marshall's lab wasn't just so much working on amazing instrumentation in the national lab, which was really cool, but also the people I got to work with were some of the brightest minds in mass spec. And so I think just through osmosis, just being around those guys, I just learned amazing things. And so it's really phenomenal. From there, I got recruited to go to a small biotech company called Allenex in San Diego. It's a combinatorial chemistry company. They got bought by Warner Lambert. And so that was exciting because the Warner Lambert, they had Park Davis at the time in Ann Arbor, Michigan. That's where Joe Liu was. And so I got to overlap with Joe Liu for a little bit. And obviously, he's a native mass spec expert. So just being around him and his group was phenomenal for also helping me learn different types of mass spec. Then Pfizer came along and took over Warner Lambert. So I was part of what was eventually Pfizer, and I was part of that whole world for 20 years. The thing I always tell people was nice about working for Pfizer is that we reorged about every 3 years. And so every 3 years, I got a new job. I started out doing combinatorial chemistry, small molecule analysis, purification, LCMS, supercritical fluid chromatography MS. And then the last group I was in was a structural biology group, mostly X-ray crystallography, but in my group, we did a lot of proteomics, HT exchange, structural biology with mass spec. And so it's all those reorgs actually made it kind of fun because I got to do a lot of different things.
So like a lot of our guests, you really have quite the journey. And I think you might win the prize for running the gamut of different fields of study, different types of analysis that you've done. So between all of these different subject areas and professional roles, what are some of the most important lessons you've learned along the way? And are there any additional people that have helped you at this time?
I think probably the biggest impact on my career was a couple things. One thing, starting out my career in biotech, my boss was Rich Griffey at the time, and he basically ran our group almost like a PhD program. And before I left ISIS, I had almost 20 publications. Um, doing everything from gas phase chemistry of nucleotides, structural analysis, native mass spec. And so being in biotech in the 90s, where you really had to learn a lot of different things and contribute to fundamental science just to show ISIS, or just to show that these different companies, biotechs, are good science companies to get funding, was a great time to be in biotech. And I think, even now, if you're in the small biotech companies, you have to learn everything and contribute to the companies to make them work. The other thing that was really nice is while I was there, I got to go as a visiting scientist to Marshall's group, as I mentioned before. And that actually came about at ASMS, my first ASMS. I met a couple of guys, Jared Rader and Chris Hendrickson. Chris Hendrickson is now the director of the Meg Lab. I met those guys on an elevator at ASMS, and we were just talking and decided to go have a beer, and we became good friends. About 3 ASMSs later, we actually talked about mass spec and what we did, and that's when I went to their lab and worked with them as a visiting scientist.
You've really given us quite the rich history of what you've done. And you've given us a little bit of a taste of some of the moments that have catapulted you into new roles. Is there a science OMG moment, even if it's not mass spec related, that really made you love the field of chemistry, science, or something?
Yep, that was so I went to Buffalo Grove High School. It's the northwest suburbs of Chicago. And I had an AP chemistry, Dr. Bowles. He was just an amazing guy, super energetic, made the class very fun, which is pretty amazing to say about an AP chemistry course. What really sealed it, it was a couple weeks before Christmas break one year. We did a saccharides experiment. And so we were mixing all these compounds together, and we made these two ropes, a red and white one, and we twisted them up. And then he told us to put a bend in them. And then everybody in the class all of a sudden realized, holy crap, we just made candy canes. And so he turned science into candy for us. And so that was, it just showed us that science could include all kinds of things, be very fun and tasty.
It's very appropriate, I think, for this last episode of the year as well as we approach the holiday season. So it was a really nice anecdote. So maybe let's turn a little bit more fully to the actual science that you've done and what you've observed in the fields that you've been in. What have you seen evolve overall, whether it's in pharma or any of the other places that you've been?
So I guess two things. So drug discovery, the advancements in drug discovery over my career have been amazing. In the way things are approached, the throughput, the speed, what a single scientist can contribute now, just due to technology, internet, even things like ChatGPT that are starting to come into everywhere now. So that's been really amazing to see. It was always, I loved working at Pfizer in drug discovery. I had friends in all different companies. The really nice thing working with Bruker now is I get to go visit these people in all the other pharma labs and biotech labs and get to talk about the science they're doing in more detail than we were allowed to talk about before. And so what everybody's doing in these labs is just phenomenal at an individual level. Mass spec wise, I guess the usability and robustness of instruments is amazing. When I first started, I was on a single quad instrument with the fusion pumps. I basically had to rebuild that instrument like every other day to keep it running, which in one day way was really great because I learned a lot about mass spec by building and rebuilding and replacing components and everything else. So that was nice. I would recommend it to a high throughput lab. So now the instruments, they're just the software, the hardware, everything about a mass spec instrument is just more usable. And now you see, 20 years ago, you would never see instruments in mass specs in biology labs. Now they're just everywhere. Every lab has mass specs. And so it's the change has been pretty remarkable.
So you mentioned a couple of different components that make up the pharmaceutical industry, but some of our listeners really may not understand or be familiar with the drug discovery pipeline. Can you tell us a little bit more about the evolution of a drug and what happens throughout that process?
Yeah. So generally, you start out, most places will start out with the disease or therapeutic area, and then you basically decide what your target's going to be. There are a lot of publications where they say, and you see like Time Magazine will say, oh, cancer is cured, we found the next target. Whether it's Time Magazine or analytical chemistry or science, it's just a constant bombardment of new targets for oncology or heart disease or whatever other therapeutic area you're working at. I mainly worked in oncology my last 10 years in Pfizer, so I'm most familiar there. But you see all these targets that are potential for, curing whatever, type of breast cancer, whatever other type of cancer you're talking about. So the first thing we do in pharma is we validate those targets. So that's very early discovery, we call in the pipeline. So there's target validation. Once a target's been validated by multiple biological, analytical, and other methods, if the target's validated, then we go on and we generally do a high throughput screening campaign. While that high throughput screening campaign goes on, there's also a lot of work with biology, which also includes a lot of analytical chemistry to figure out mechanism of action. If mechanism of action can be defined, chances of success in clinical trials are increased dramatically. And then the next stage is you move down the pipe, you get a lead compound, and then you start looking for biomarkers, things like that. Having good biomarkers also increases your chance of clinical success quite a bit. As you move down, we call it moving downstream, you get into the preclinical, call it pre-human studies. Then you move to human studies, phase 1 trial. That's generally just as the drug tolerable to humans. And then you move to phase 2, phase 3, where you look for efficacy, and then the drug hits the market.
So, Mike, how does mass spectrometry fit into this? What stages use technology as critical aspects? Are mass spectrometers important in pushing this to clinical trials and into eventual approval?
The fun thing about working in mass spectrometry is that it kind of applies everywhere. And it's also really nice because it doesn't just apply to drug discovery. Mass spec can be used for petroleomics, for food science, for looking at toxic waste, all kinds of different things. In the drug discovery pipeline, mass spec is used basically everywhere. It's used in the very early discovery stages, especially for target validation, things like that. It can be used for screening. The Bruker timsTOF is being used in lots of early discovery labs for basically every part up to preclinical trials. When you get farther downstream, mass spectrometry, usually coupled with LC, is used for QC, could be used for QC of the drugs, could be looking for impurities in drug product. And then it's also used in later stages looking at drug metabolism. You get blood samples from clinical trials, and there you're going to start also looking at drug metabolism, and you can be looking at things like biomarkers, things like that. We do have good mass spec imaging with our Bruker instruments, and there you can take a look at drug distribution. And really importantly, we can look at non-targeted approaches. And so if there's an unknown metabolite, we can identify those too.
So, with all of this and all of the technology that goes into pharma, what do you see as one of the biggest barriers for mass spec to be marketable to the pharma or biopharma industry?
Well, mass spec is already very marketable to the to the pharma industry. Literally, almost every single pharma lab you go into, there is going to be a mass spec doing something. To increase that, it just basically comes down to ease of use and software. when you go into, you have your core mass spec labs where the guys, the people in the lab are kind of gearheads and want to do everything with the instrument, do a lot of exploration. But then you have, maybe a recombinant protein lab where they're just making proteins for project teams, and they just want to look at and get a accurate mass of the intact protein. So for those labs, they want to be just a walk-up instrument. And there are already, lots of walk-up instruments for chemistry and biology. And so easy use is there. Now it's down to, can we make these more complex experiments easier to do and reproducible? And most common types of experiments now are already there. And now we're really pushing into realms where we're looking at like top-down sequencing, and top-down sequencing out of biofluids, looking at really complex samples. And so as the complexity of the samples increases, that's where we need to have a few more innovations to help clean things up.
You just picked up on one of the words that I want to also highlight. You work for Bruker, I work for Bruker. Now that you're on this side of it, can you explain how the mass spectrometry evolution occurs and what the timeline is there? We can't put a new product out to market next week. So how long does it take for innovation to occur in our field as well?
So I guess there's two kinds of innovation. One is instrumentation. If you look at just the hardware and what hardware can do, it's really, really amazing. A perfect example is, Bruker. We just purchased Asmatech at the beginning of this year. They make something called an omnitrap. It's a fairly complex component to the mass spectrometer, and it can do really amazing things. And so the idea is we take that and we click it onto one of our instruments, and we have a new instrument. And so that's relatively easy to do in a breadboard fashion, but to have it as a robust instrument is just an amazing amount of work. And it's not just the hardware, it's the software too. So there's the hardware component. I think those innovations are just happening all the time, but it's translating that into a robust instrument with software to back it, the ease of use. That is really where the complexity comes in, and the product development really requires a massive amount of teamwork. You can have the best physicist in the world, but if you can't make something where a pharma scientist sits down in front of the computer and can't figure out how to turn it on, then it's no good. And so the teams to build these products and bring them forward to market are extremely diverse and have to be, everybody on the team has to be really knowledgeable and aligned with what they want to do. So it's something now working for Bruker as opposed to working for Pharma. I have a whole new appreciation of not just product development, but the whole sales cycle and everything else that goes behind it.
So what about post-sales? That's one of the things that you didn't touch on. What's something that mass spec companies do to help people get up and running? And what does that look like from your perspective?
So one of the things when Bruker started the Pharma Biopharma business unit, which I'm directing, they started the unit 6 years ago. One of the first things I said is to differentiate ourselves from the rest of the vendors is going to be support of our customers. As I said, the hardware out there is really, really amazing. For the most part, pretty robust. There's great instrumentation. I believe at Bruker we have some really amazing, unique things. But again, none of that matters if you can't support your customers. So one of the things we do is we have field ask people just to sign to pharma labs to understand the pharma language. And we'll go in there and help people, sit with them as long as they need to get an understanding of how to do the software, develop methods, and operate. So I think really post-sales support is a huge thing. When we talk about success in a pharma lab, it's not selling instruments. It's actually having that instrument being used on a daily basis. Selling an instrument and having to sit in the corner, to me, is pointless. we want it to be used every day. And when it's used every day, of course, they're going to buy more, which is good for us.
So, Mike, it's been really great chatting with you. I always enjoy hearing about the professional path that people have taken. But I know you have another major theme in your personal life, and that's of sports. You played college football. You're currently a very avid hockey player as a hobby. Can you tell us how sports has provided you with a youthful mentality towards your career and how it's influenced you?
Yeah, I think a couple things. One is teamwork. It's interesting when I interview people, I can generally tell if they played a team sport sometime in their life. It just kind of comes across sometime. One thing I had no clue about, I spent my whole career in basically working in the lab, doing biotech and pharma, drug discovery experiments. Didn't know anything about business, didn't know anything about sales, marketing, anything like that. I was very fortunate. Rowan Facura hired me at Bruker and basically taught me about all of these different things. And one thing I had no idea is that sales, especially the way we do sales for pharma, is probably requires more teamwork than anything I've ever been involved with. A lot of people think it's just the salesperson goes out there and say, "Hey, buy some stuff," and that's it being done. But within Bruker, we have business units, we have marketing people, we have the software teams, we have the hardware teams, the service teams, and we need every single one of them involved to make a sale happen. And within the pharma group, we view ourselves as a global team. If you look at any of the big pharmas, they have labs around the world. So the pharma team at Bruker, we're a global team. We interact across the globe constantly to make sure we're aligned. And by doing this, it really makes us more effective. And so I guess I spent my, kind of early life doing sports on teams, and I see how important it is in the sales process too. And I try and bring that into the Bruker world. So yeah, there's the team aspect, which is pretty obvious. I guess the other thing you learn in sports is that you learn different ways to push yourself, push other people, and just to fight through whatever problems there are. Our job as scientists is generally problem solving. One of the things, you learn in, chemistry or whatever science classes you're taking is the scientific method. And, the scientific method actually is basically what you apply to any, like a sports situation at halftime when you're trying to solve problems. And so you're applying things like that. You're pushing yourself and, bringing everybody in, every component in you can to help achieve the goal. So I guess that would be the second thing. It's kind of like the first thing, but a little different.
So I may not like the answer to this, but we've known each other for a few years now. Do you think that I played sports or not based on the interactions that we've had over the years?
I would say no.
You're correct.
And it's not a negative thing. It's just a different way people approach situations. And I've had people in my group who are just really been outstanding scientists who never played sports. I encourage them as adults to go play. No, you can still learn.
Don't worry, no offense taken. All right. Well, I think with that, I'll say thank you once more and appreciate your insight and your thought leadership in this field.
Thank you. It's great talking to you.
Welcome back to OMG OMX, and I'm really thrilled that you've stuck with us throughout the year. If you didn't know this, only 10% of podcasts make it past 3 episodes, so I feel like we've done pretty well this year. Thanks for staying tuned to listen to Mike Greig.